Hello learners, in this video we're going to be going through your AQA A-Level Biology paper 2. So hopefully you've done paper 1 and paper 3 already, but don't forget any of the topics that we think might come up on paper 2 can also come on paper 2 or on paper 3. So go through this, use this preparation for paper 2, but don't forget about this for paper 3.
ATP production occurs in both photosynthesis and aerobic respiration. Complete the table below to show which statements describe each process. For each statement, place a tick in one column, both columns, or not at all. So the first one, it says electrons pass between protein carriers. So this is just the electron transport chain. So both photosynthesis and respiration have an electron transport chain, so we need to tick in both. Next row says electrons are excited by light energy. Only photosynthesis involves light energy, so then we're just going to tick the photosynthesis box. The next one says it involves chemiosmosis. So this is just creating that chemical gradient of H+ ions, which is then used to make ATP through ATP synthase. That's what chemiosmosis is about. Both processes produce ATP in this way. And this is all about ATP production in total between the two. So yes, the production of ATP in both involves chemiosmosis. Includes photophosphorylation. So again, the word photo here is suggesting of light. Light energy is what is used to drive that phosphorylation, which helps to produce ATP in photosynthesis. And so yes, this is just photosynthesis, it's not respiration. Oxygen being the final electron acceptor, that means oxygen is needed or used in the reaction, and we should know that oxygen is the final electron acceptor at the end of the electron transport chain in respiration only. Okay, we don't need oxygen, we don't put in oxygen into the process of photosynthesis, it produces oxygen at the end, so that is respiration only. And then lastly, involves the matrix and the cristae. So these are both structures we should recognise as being in mitochondria, and obviously the only process here that uses mitochondria or takes place in the mitochondria is respiration. Okay, so you get a mark for each column being correct. So it does mean that you could potentially have ticked some of them wrong, say for respiration, but if the photosynthesis column is all correct, then you can have 1 mark. The Krebs cycle is needed to produce a large amount of ATP. State where the Krebs cycle occurs. So we don't need to do anything about this diagram for now because it hasn't asked us to look at anything, we just know that it's a diagram of the Krebs cycle. So where is the location where this stage of respiration happens? We should hopefully know it takes place in the mitochondria, but specifically the matrix. You aren't going to get away with saying mitochondria as your answer on its own because we are now at A-level stage. We know a bit more detail about this. We know exactly where in the mitochondria the Krebs cycle takes place. It takes place in the matrix. So if you just put matrix, you get a mark. If you just put mitochondria though, you do not get a mark. Explain what happens to the reduced coenzymes after the cycle is complete. So it's talking about reduced NAD and reduced FAD. So what happens to these after the Krebs cycle? It's an explain question, but it's asking about what happens to them. So where do they go, but explain why they go there. So they travel to the electron transport chain to release their H+ ions and electrons. This obviously helps to produce the ATP through the electron transport chain through phosphorylation. So this is why they go and where they go. They travel to the electron transport chain to release H+ and electrons for phosphorylation. You don't need that part on the end for phosphorylation, but we need to make sure that we're saying that either we talk about them being oxidized or them releasing those H+ ions and electrons that we need in order to do the next part of respiration. Give the name of the molecule or molecules represented by the letters in the diagram. So now we're looking at this diagram properly. We've got a diagram of the Krebs cycle. Obviously some bits are missing. They're represented by letters. We have to figure out which thing goes in these gaps where the letters are, and it could be more than one molecule. So the first part is something that's coming into the Krebs cycle. So what's coming in at that start point, it's acetyl-CoA or acetyl coenzyme A, which we hopefully know comes from the link reaction. So for B and C, they're both at the point where there's a loss of carbon. Okay, so there's a loss of carbon from 6 carbons to 5 and then 5 carbons to 4. This is the stage of the cycle that we call decarboxylation. So carbons are being removed, and this is the point then where those carbons then end up being carbon dioxide, and this is where the carbon dioxide from respiration is being released. And then finally D, we've got an arrow from D to ATP, so it's whatever is in D is being used to make or produce that ATP. So what is ATP made of? It's made of ADP and inorganic phosphate, or PI. So this is why they said molecules in the question, because obviously that's 2 as opposed to all the others being just 1. So acetyl-CoA for A, B and C are both carbon dioxide, and then D is ADP and PI, or inorganic phosphate. The rate at which the Krebs cycle proceeds is carefully controlled. If ATP begins to accumulate, the rate is slowed down. This occurs because ATP acts as a non-competitive inhibitor of the enzyme isocitrate dehydrogenase 3, or IDH3, which produces the first reduced NAD in the Krebs cycle. Explain the mechanism through which this allows ATP concentration to control the Krebs cycle. Okay, so it tells us that ATP acts as a non-competitive inhibitor for this enzyme, and it tells us that the enzyme creates this first stage of the cycle where NAD is reduced and we get the kind of loss of carbon dioxide and we get to that 5-carbon molecule. So a non-competitive inhibitor, so this is really an enzyme question as well as a Krebs cycle question. So non-competitive inhibitor means it binds at the allosteric site, or it binds away from the active site, but it changes the shape of the active site so the enzyme no longer works. So if this doesn't happen, if we don't get this first part of the reaction for the Krebs cycle, then what's going to happen? Well, we won't get the 5-carbon molecule made and the cycle is going to stop. So it tells us that if ATP accumulates, then the rate of the Krebs cycle is slowed down, which ultimately will mean less ATP is going to be made because there's going to be fewer coenzymes going to the electron transport chain. And if that happens, so ATP is getting used up, and therefore it's going to decrease in the rate, then we're going to then get less inhibition of the enzyme and it could speed back up again. So this only happens when there's lots of ATP accumulating, and then this inhibition occurs, and then it slows down the Krebs cycle, which slows down the rate of ATP production, and then ultimately that will reduce the amount of ATP, and then the inhibition will reduce, and then we'll go back to the rate of the Krebs cycle speeding up again. So really speaking, that's what it's asking. So we're kind of talking about what we know about the non-competitive inhibitor, then say what happens then if that inhibition happens, what do we not get? Link that to less ATP being made overall, and then we can sort of link this idea of control to the fact that that will mean then there's less inhibition of IDH3 and we can start increasing the rate again. So ATP binds away from the active site of IDH3, causing the active site to change shape. IDH3 can no longer bind to the substrate, so no 5-carbon molecule is made in the cycle. The cycle stops or slows down, so less ATP is produced as fewer coenzymes are produced. Eventually, the ATP concentration decreases until no longer inhibiting IDH3. Students wanted to investigate the natural behaviour of woodlice. They set up a choice chamber as shown in the diagram below with 4 different environments. They added 10 woodlice to the central point marked with a cross. They left the woodlice for 10 minutes and then recorded the number of woodlice in each section. They repeated this 4 times and calculated a mean. So hopefully we recognise this as the required practical for animal responses. Explain the purpose of the silica gel. So why do we need to add the silica gel to sections A and B? Well, it says we're creating 4 different environments. So we've got dark and damp, light and damp. Then we've got dark and light on the other side for A and B, but we've got the silica gel. So what's the opposite of damp? It's dry. And if you didn't know or haven't used this before or haven't heard of it, that would be how to work it out. Basically, it's the opposite of what's in C and D to try and create 4 different environments that can be compared. So silica gel comes in those kind of little packets. Sometimes you might see where it says like, do not eat this, and it tries to dry out boxes or packaging materials. So it absorbs moisture or it reduces humidity in order to create a dry environment. Suggest 2 steps the students should have taken to ensure appropriate handling of the woodlice throughout the experiment. So this is about being safe and ethical when we're handling organisms for these experimental practicals or practicals with animal responses. So there's a couple of answers, sort of 2 spring to mind for me would be about how to be gentle when handling them, so not causing them any damage, so using like a paintbrush or a spoon, something that's soft, not sharp, not forceps, nothing that you're going to damage them with. And also when you're using the organisms, making sure that if you're going to sort of take them, you're putting them in this new environment to try and put them in a choice chamber or a maze, just making sure they're not out of their suitable habitat for longer than is necessary. So they're obviously like dark, damp conditions. You could have them in a tank with some soil or some bark or something like that. And that's something you want to be spending most of their time in and then taking them out for short periods of time to put them in the maze. You've got other options in the mark scheme, even simply things like kind of washing your hands before and after using them or touching them, and just making sure you're kind of being aware of how you're handling and using them. So I've gone for using a paintbrush to move them to reduce damage and replacing woodlice into dark, damp habitat when not using, e.g. between repeats. Suggest a null hypothesis for this investigation. So hopefully we know that a null hypothesis is always kind of a no or a negative, what you are not expecting to happen, so that there's going to be no difference in this case between the number of woodlice in the different sections. So we are assuming that because we've got the different environments and we know that woodlice prefer that dark, damp environment, we're assuming there's going to be more of those in that environment when we've left them after 10 minutes than there would be in the dry or light environments. So therefore, our null hypothesis that won't happen is that there will be no difference in the number of woodlice in each environment or each quadrant or each, whatever you want to say, section after 10 minutes. The table shows the students' results. The students conducted a chi-square test to determine the significance of their results. Complete the expected row of their results table. So remember, we've said there should be no difference. Our kind of null hypothesis would be that there's no difference between the number of woodlice in each section. And so for this, that would mean it would be exactly the same in each section. We use 10 woodlice in total, so we need to divide it by 4. So for the purpose of the chi-squared, we would say that we would expect to see around 2.5 woodlice in each section after 10 minutes. And then obviously what we're doing is we're comparing what we actually saw to that when there would be no difference between them. The student's final chi-squared result was 8.4, and this gave a p-value of 0.038. Explain if the student should accept or reject their null hypothesis. So anytime we see a p-value, we have to think about whether or not that p-value is less than 0.05. If it is, in which case this is, then there's less than 5% chance or less than 5% probability that the result was due to chance. So this means that it is significant because something is causing this difference. Therefore, we reject the null hypothesis. The p-value is less than 0.05. So this means that there is less than 5% probability that the result is due to chance, so they can reject the null hypothesis. Remember, this is exactly the language we need to use every single time we do a statistics question. If you're answering a question about a p-value and you're not writing in this way, then there's something wrong. Obviously, you would change it if it was greater than 0.05. You would say there's more than 5% probability, so it's not significant. But other than that, the language should be exactly the same. Explain how the reflex behaviour of woodlice explains the students' results. So I've brought over the results table and the diagram so that we know what we're looking at so that we can easily explain what's going on. So A is the dark, dry environment, B is the light, dry environment, C is the dark, damp environment, and D is the light, damp environment. As you can see, there's clearly way more woodlice in the dark, damp environment in C than the others, which explains our significant result. And clearly this is because woodlice prefer to be in that dark, damp environment. So when we're answering this question, we've got to remember that we should be mentioning how and what explains their behaviour here. So we know they prefer to be in this environment because that's where they live, but how did they end up here? How did more of them end up here after 10 minutes? It's because of kinesis, it's a kinetic response. And we need to think about the fact that we know that this practical is about animal responses, so we know that we should be mentioning something about that type of response in our answer. Greater numbers of woodlice in dark and damp habitats suggest that through kinesis, they stayed in their more favourable habitat after 10 minutes. So they would have moved randomly through the maze, but obviously would have moved more rapidly when they were in the dry or the light environments. And then once they found the dark, damp environment, they would move much more slowly or reduce their movement. So they ended up staying in there for longer. The diagram below represents the stages of the nitrogen cycle. Name the type of bacteria carrying out the process occurring at A and B. So at A, we're taking nitrogen gas in the air and converting it into a form that can be found in plants in terms of amino acids and DNA. So that must mean that they are fixing nitrogen fixing bacteria. And then B, we're taking ammonium compounds and then converting them into nitrates in the soil. So that is done by nitrifying bacteria. These are just, we need to just know what each type of bacteria in the nitrogen cycle does and then be able to spot that by interpreting a diagram. Describe how ammonium ion concentration increases in the soil when organisms die. So we're looking at this part of the diagram here where we've got kind of soil bacteria or microorganisms, which we know as saprobionts. They are breaking down nitrogenous waste in this case, but we are obviously talking about dead material as well. And through the process of decay, by digesting proteins and amino acids from the waste or from an organic dead organism, then they will be able to release the ammonium ions. So this is what we're saying is happening. So we've got to link this idea that we're taking proteins and amino acids and we're digesting them, broken down by saprobionts who are doing this process of decay in order to then release the ammonium ion concentration. So you may have learned this process as ammonification, which is how I like to kind of remember it. So that's what the idea of decay is, and the role of bacteria and microorganisms, in fact fungi, into the nitrogen cycle is through ammonification. Explain what soil condition can cause process C and why it's a problem in agriculture. So process C is where nitrates in the soil are being converted back to nitrogen gas in the air. So this is clearly denitrification. It asks what soil condition causes this. It is when the soil is in an anaerobic condition, because these bacteria only work in anaerobic conditions. And why is this a problem in agriculture? So why is denitrification an issue? Because it's removing nitrates from the soil, and therefore there's less for plants to use for growth. And that's something that obviously agriculture relies upon these plants growing really well in the soil. Anaerobic conditions, which cause denitrification, which removes nitrates from the soil, so there is less available for plant growth. So it feels like a lot there, but we really need to give the anaerobic conditions as one mark, and then it doesn't necessarily say we have to name process C, but we should know that it's denitrification and why that's an issue. Farmers across the UK plant leguminous crops to increase the fertility of their soils. One farmer is choosing between two possible legumes to plant in their fields. They want to plant 500 individual plants in one field. So my first thought, and the first thing I'm going to write down before I even read the question, is that leguminous crops are the ones that are able to do nitrogen fixing because they have the association with the bacteria. And when it says fertility in soils, they want to increase the amount of nutrients, specifically nitrogen in this case, obviously into the soil. Use the information in the table below, as well as suitable calculations to evaluate which leguminous crop would be more beneficial to the farmer to plant. So suitable calculations, we need to use some maths, use some calculations to show which one of these do we think is more beneficial, is going to be more beneficial. The idea is obviously he's planting 500 individual plants of each. So if he was to plant 500 chickpeas versus 500 lentils, what would he get out of it? And the idea of planting a leguminous crop, as we said, is that it fixes nitrogen. So which one is going to increase the nitrogen the most in the soil? And then also now we have to factor in cost. So for that, which one's going to be the most cost effective, but potentially also make sure that we get as much nitrogen in the soil as possible. It is an evaluate question, which means we have to kind of look at pros and cons. So the chickpea is the cheapest per plant. It will cost, almost 50% less to plant the chickpeas versus the lentils, because it's £1 versus £1.50 per plant. There's going to be more water needed though, so that's a con for the chickpeas. We are going to need to use more water per day in order to grow them. But the soil nitrogen is double for the chickpea compared to the lentil. And although the biomass is larger, the percent of biomass that is nitrogen is actually lower, which means that although it doesn't look like a lot of the nitrogen is being fixed by the bacteria, more of that nitrogen that's being fixed by this plant is ending up in the soil than it is in the plant biomass. So we can do some calculations to show that although there's a lower biomass for lentils, a greater percent of nitrogen is going to be trapped in that plant biomass, which is fine if you are then going to plough these plants into the soil at the end, potentially, and let them decay. But if you want to try and sell this as a profit crop and still make sure that there's enough nitrogen left in the soil for your next crop, then chickpeas is going to be better. Because if you look at it, if you do some maths, if you do 400 times 0.022 versus 500 times 0.015, so we get the percentage of the biomass that is nitrogen for each one, you've got more biomass in the plant for the lentil than for the chickpea, and you can see that explains why there might be more soil nitrogen left over. So the farmer should plant chickpeas as they'll be £250 cheaper to plant. Okay, so we've done a bit of maths there. And although they will need more water, they will double the amount of soil nitrogen. Compared to lentils, as less of their biomass will contain nitrogen. And then I would literally put the sums in that we've just done up here. So that shows your maths, it does some calculations. You've shown that you understand that farmers are going to want more nitrogen in the soil. That's the whole point of this. And actually you can get that with a cheaper plant, which is chickpeas, even though he will have to plant or use more water per plant per day, but it's only 1 litre more per day. Planting leguminous crops can help reduce the need for farmers to use inorganic fertilisers, which can lead to the death of organisms in local bodies of water. Explain how. So this is just a question about eutrophication. So if we think about the brief stages of eutrophication that we have to be able to make sure we can write out here. So we have an algal bloom as a result of that extra increased fertiliser in the water. That blocks the light, which prevents plants inside the body of water, like at the bottom of the pond, for example, from doing photosynthesis. So those plants die. Then decomposers use up oxygen that's in the water in order to decay those plants. And obviously, as those plants are dead, they're not going to be adding any more oxygen to the water through photosynthesis, so the oxygen just gets used up until there's not enough there to support any other organisms. So we can write this out in about 4 sentences. Inorganic fertiliser in the water causes algae to grow rapidly. These block out the light, and plants can no longer photosynthesize, so they die. The decomposers feeding on the dead plants use up oxygen through respiration. Eventually, not enough oxygen in the water to support organisms. So I've done 5 sentences, but actually I think that last one wouldn't necessarily need that in order to get all of the marks. Heather plants are large shrubs which grow on moorland. Some can be seen in the picture below. Landowners often control the growth of heather by burning these areas of moorland. State the term described by the following statements. What is the type of succession which occurs after the burning of moorland? So by type of succession, it means either primary or secondary. After the burning of the moorland, there'll still be soil. So when we're not going from bare rock, nothing, it's going to have soil, it just be kind of starting again from there with the plant life, so it is secondary succession. What's the type of community produced when full succession is diverted or prevented in this way? This is a kind of another way of saying deflected. So this asking basically what happens, what type of community is formed when we deflect succession, so we're not allowing it to go to the full climax community. What type of community then do we form? And that is known as the plagioclimax community. Explain why farmers may want to periodically burn the moorland to remove the heather. So the clue here is farmers. So why would farmers want to burn moorland and remove the heather, which is obviously the large shrub plant that mentioned in the start? So they're going to want to graze livestock on this moorland. They're not going to want to grow crops or anything because it's sort of hilly, like in the picture. It's describing and showing you that it's not kind of a flat field, but they could graze their animals there. So by periodically burning it, we've just said, what does that do? It creates that plagioclimax, so we prevent those large shrubs from growing. And that's obviously going to mean that there's more kind of smaller, low level plant material that the livestock can eat. They're not going to eat big shrubs, and also it means it's sort of a bit more open so that they can get and maneuver around these big shrubs. So you've got kind of two options here. You can either talk about having new, fresh plant life growing up from the ground, that then there's suitable amounts of that for them to eat, or just prevents the large shrubs from growing and getting in the way so that the land is useful or suitable for grazing livestock. The table shows the changes that occurred in one area of moorland after burning left bare ground. Using evidence from the table, explain how succession has taken place on the moorland. So we need to just really describe the steps in the process of succession, but we need to use the specific sequence of what happened to the plant species in the table. So we can see that over the time, the mean percentage of heather has increased, but the number of other plant species has changed. So we've got initially quite a few, not very many species, then a big increase between 0 and 4 years, then a decrease from 40 to 15. So we've got a big increase and then fewer and fewer as the percentage of heather increases or becomes more dominant. So we have very few plant species to start with as new species colonise. Then obviously they make the conditions more favourable or more suitable by dying and decomposing and increasing the nutrient amounts. And therefore that's where we then get that big increase in the number of different plant species, including some heather to start with after year 4. And then we see a decrease. So that decrease as the amount of heather plants increases and we get that kind of domination of heather, it's going to cause that more hostile environment. It's going to outcompete the small plants, shade them potentially, and therefore they're going to find it harder to survive around the heather, which is why we go down to a decrease of less than 15. So we've got to talk about the initial start, then the big increase, and we've got to explain why that happens as well using our knowledge of succession, then talk about the slow decrease and explain again why that happens due to a knowledge of succession. So here's my kind of answer. I've got all the mark points, not necessarily in the same order that it is in the mark scheme, but they are there. So 1 mark for talking about colonising. So initially there are very few plant species, as only a few specialised species will colonise the bare ground. Then after 4 years, there is a large increase in the number of different plant species and some heather present, as the pioneer species improved the soil nutrients, as the death and decomposition of the initial colonisers made the abiotic conditions more favourable for more species to grow there by increasing the nutrient availability and stability of the soil. So that whole sort of second part of the paragraph gives you kind of marks 2, 3 and 4, because it's talking about the big increase, but it links back to the decomposition of the colonisers, the pioneer species, making the nutrients increase, and then it also links to that kind of nutrients and stability, which is the reason why there's been this big explosion and expansion of species after 4 years. Then we've got between 9 and 12 years, there's a decrease in the number of different plant species and an increase in heather. as heather becomes more dominant species and makes the environment more hostile to smaller plant species by outcompeting them for space and light. Eventually, there are very few other plant species that can survive, and heather covers the majority of them all. So there we go, we've got those kind of last two mark points about how the heather increases whereas the other species decrease between 8 and 12 years, or 9 and 12 years. And then we've got this idea of like that dominant and outcompeting and making the conditions more hostile, competing with other species, which is why it becomes more dominant. So there wouldn't be very many marks here if you just wrote about succession without relating it to the table, because that's what it wants you to do. So you wouldn't get very few marks at all if you just talked about pioneer species and just went through kind of a standard set of bullet points. This is one of those ones where you've really got to apply what you can see in the data and link it to your knowledge. The diagram shows a neuron. Give the name of the cell that forms part X and explain how it helps the neuron to carry out its function. So be careful here, it wants the name of the cell that forms this part, and then it wants us to explain how that part X is used to help the neuron carry out what its function is, which is to transmit electrical impulses. So hopefully we know that the name of the cell that secretes what is the myelin sheath is a Schwann cell. And obviously then we're being asked to talk about how part X, so how the myelin sheath then helps the neuron to carry out its function. So it insulates the axon, so it allows saltatory conduction, which obviously speeds up the impulses transmission, so it's faster. So we've got this idea, we've got to add the cell for 1 mark. What does X do? And how does it help it carry out the function? So it insulates, and how does that help it do it? It obviously increases the speed of conduction. So a Schwann cell produces the myelin sheath, which insulates the axon, causing saltatory conduction, which allows transmission of impulses faster, or increases the speed of impulse transmission. The diagram shows the changes in membrane potential during an action potential. The changes are due to the movement of ions across the membrane of the axon. Explain what causes the change in membrane potential between points A and B. So we need to give reasons for the change, and the change in potential goes from about -65, -70 up to +45. So it's just asking us to explain why the membrane becomes more positive, or why depolarization happens. So hopefully we know that it's because the voltage-gated sodium ion channels open, and therefore sodium ions diffuse into the cell or into the axon. So that influx of positive ions increases the charge on the membrane, it becomes more positive. So just be careful when you're writing answers to this, that you're saying sodium ions. So either you're writing the word sodium and ion, or you're writing Na+, that's fine, but you can't just say sodium, because it's not sodium channels and it's not sodium diffusing in, it's sodium ion channels and sodium ions diffusing in, and that bit is important because it has to have a charge, because otherwise we wouldn't get depolarization. Name the stage between C and D. So this is a point where the membrane is returning to resting potential, and it's the point at which there can be no new action potentials until after D, and we're back at maintained resting potential. So this helps to create that gap between action potentials to make sure we don't get, like, they don't blur into each other and they're separate, and this is known as the refractory period. Hopefully we know that. When an action potential reaches the end of one neuron, it must cross the synapse. The average gap between two neurons is 32 nanometers, and the average time it takes for the impulse to pass between neurons is 1.6 times 10 to the minus 7 seconds. Calculate the speed of the impulse crossing the gap between two neurons. So speed equals distance divided by time. This is a physics equation that we should hopefully remember from GCSE. It's something we should know how to work this out. So we've got speed, we've got distance, easy peasy, other than the fact that we are going to have to convert distance, because if you remember, distance is always in meters and time is always in seconds, and we need to give our answer in meters per second. So this is just a maths question. It's about SI units. Do you remember a bit of physics from GCSE as well? So we've got nanometers to micrometers, micrometers to millimeters, millimeters to centimeters, and centimeters to meters. So hopefully we know these conversions. And then once we've figured out, so nanometers to micrometers is divided by 1,000, micrometers to millimeters is divided by 1,000, millimeters to centimeters is divided by 10, and centimeters to meters is divided by 100. If you add up all those zeros, there's 9 zeros there. So we effectively need to convert 32 nanometers into meters. We're going to need to divide by 10 to the power of 9. So that gives us 3.2 times 10 to the minus 8 meters. And we can now just do that divided by our time in seconds to get our speed. So our speed of transmission is 0.2 meters per second. Parkinson's disease is a neurodegenerative disorder in which patients find information processing, attention, and memory difficulties. In patients with Parkinson's disease, the speed of transmission of impulses across synapses, which use the neurotransmitter dopamine, are reduced. Suggest why Parkinson's can be treated with drugs that brain neurons can use to make dopamine. So in Parkinson's, it's saying that the speed of transmission is reduced in neurons that use dopamine. It's asking us to think about why having drugs that increase the levels of dopamine by allowing the neurons to make more of it might help treat Parkinson's. So why does increasing dopamine increase the speed of transmission? Because that's ultimately what we're being asked to do here. So if we think about dopamine, the fact that it's a neurotransmitter and what a neurotransmitter does, it's going to diffuse across the synapse. We've just been talking about the synapse. So this is the idea that the speed of transmission between one neuron to another is too slow, and that's why they have some of those issues in Parkinson's. We're giving the neurons the ability to make more dopamine in the neuron, so that means there'll be more of it in the presynaptic neuron, which means more of it can then be released into the synaptic cleft, and there'll be a greater concentration gradient between where there is a lot of dopamine because it's just been released at the presynaptic bulb, and not a lot of dopamine by the postsynaptic membrane, so it's going to diffuse faster if there's a greater concentration gradient. If you're not making and producing and releasing as much dopamine at the presynaptic neuron, then there's going to be a lower concentration gradient, so it's going to diffuse slower. You can also just argue that if there's more dopamine, more of it will diffuse across, and therefore more will bind to the receptors, and therefore it's more likely to trigger an action potential. That's also a mark here as well. My brain went to diffusion across the synapse and then went to concentration gradients. But if yours didn't and you just talked about there being more, then that's also fine for a mark. If neurons can produce more dopamine in the presynaptic knobs, then there'll be greater concentration gradient of dopamine. Between the pre- and postsynaptic neurons. This will increase the speed of diffusion of dopamine across the synaptic cleft, which will increase the speed of transmission. Okay, so the main key things here are talking about the fact that the dopamine is going to be being made and released from the presynaptic neurons, because that's showing our understanding of neurotransmitters and how a synapse works. And then we've got to link this idea of more dopamine. Why is that going to create faster transmission? The diagram shows an ocean food chain and the energy transfers between trophic levels. Name an essential group of organisms that is missing from this food chain. So we've got phytoplankton, zooplankton, herring, and yellowfin tuna. The group of organisms that's missing, so it is a group, so it's not a species, it's not even necessarily saying it's a trophic level, but the way these food chains would kind of cycle back would be through decomposers. So decomposers are part of food chains because they complete them. So when any organism dies or there's any waste material from these organisms, that is obviously broken down by decomposers and recycled those nutrients back so that phytoplankton or other producers at the start of food chains can then reuse that biomass and start again. So it is decomposers. State the equation that links gross productivity and net productivity and use it to explain why not all biomass is passed to the next trophic level. So gross productivity is GP, net productivity is NP. So hopefully we know this in the equation is NP equals GP minus R. What does the R stand for? Well, the R stands for respiration. So this explains that net productivity is what is left over from the gross productivity minus the biomass that's used for respiration to provide energy for all the living processes. So we need to give the equation and then explain that some biomass is used in respiration. In this ecosystem, the gross productivity of the zooplankton is 24,525 kilojoules per metre per year. About 5.7% of energy in the herring is used to produce new biomass. Calculate the energy stored in the new tissues of the herring. Give your answer to two significant figures. So the gross productivity, or GP, so that's like the total biomass produced by the zooplankton in a year, is that big 24,000 number. Then 5.7% of energy in the herring is used to produce new biomass. So 5.7% of what the herring takes in from the zooplankton is going to be used to make new biomass. The rest is obviously going to be used for respiration, released as heat energy, used for movement, etc. So they only take 12.7% of that productivity from the zooplankton, and then they only use 5.7% of that 12.7% to actually produce biomass. So we need to do 12.7% of the big number and then 5.7% of that 12.7% to get the answer. So this is like we're basically calculating the net productivity of the zooplankton and then the net productivity of the herring effectively. So we're going to do 24,525 times 0.127, and that gives us 3,114.675. I wouldn't round any numbers when you're doing stuff like this until the very end, just to be on the safe side. So we then take that number and we do times 0.057 to get the 5.7%, and that's going to give us 177.53. It wants our answer to two significant figures, so we have to round all the way up, so we get two numbers and then a zero to get that to two significant figures. So the closest whole number that we're going to get then is 180. Phytoplankton live in the upper parts of the ocean, and large blooms occur between March and May at the start of spring. The biomass transfer efficiency is the highest during a bloom. Suggest why. So the transfer efficiency is going to be higher because more biomass is there to be able to be transferred. And it says it's highest during a bloom. It tells you a bloom, or we should hopefully know that a large bloom is where there's lots of growth of these plankton in one go. And the phytoplankton obviously produces, and it's saying that the bloom happens at the start of spring. So why is there a bloom? Why is there greater growth? Why is there therefore more biomass? And so thinking about the things, if they're living in the ocean, they say they're living in the top of the ocean, they're producers, which means they must absorb light energy. At the start of spring, it's going to be warmer, it's going to be sunnier. So I'm going to go with the fact there's more light and therefore more photosynthesis. This is one of those questions that suggest questions, so it's suggesting how you use your knowledge and sort of the bits of information that's being given to you from the question. If you know there's producers, you know there's tons of them growing in the start of spring. You've got a lot of information there to kind of lead you to this idea that more light, more photosynthesis, and therefore there's more biomass being produced in order to transfer it. So the light intensity increases, so the rate of photosynthesis increases and more biomass is produced. You've also got the option to do temperature. So it's warm and sunny. So I went for sunny, but you can go for warm and talk about the increase in temperature, increase in the rate of photosynthesis still, because that's the reaction that's going to produce the biomass in the producers. Some stocks of yellowfin tuna are being overexploited. Explain the impact of this on the zooplankton population. So this question is basically saying what will happen to the number of zooplankton if the numbers of tuna decreased, and why? So what would be the reason? So the herring, there would be more herring. So always start from the top and work your way down with these questions. So there would be more herring as there would be less tuna to eat them, so there'd be less of the herring being eaten, so there'd be more of them surviving, so their numbers would increase. That would mean that there would be less zooplankton because there'd be more herring to eat them. So more of them being eaten because there's more herring. And therefore, our answer is what will happen to the zooplankton. Their numbers will decrease. Why? Because more herring are going to be eating them because less herring are being eaten by the tuna. So there's obviously a few ways you can write this. It's quite simply done in the mark scheme. So I'm going to write it as I would explain it. So the number of zooplankton would decrease because then we know we've definitely got that mark for that first point. There'll be more herring to eat them because there are fewer tuna eating the herring. And I've added that last bit because that makes sense to me and that explains how I understand what I'm talking about, but probably wouldn't necessarily need that last sentence to actually get the second mark. The data in the graph below shows the annual mass of tuna caught and the biomass of spawning reproductive age of fish in the population since 1950. So we've got two axes here. We've got the total number or the amount of biomass of fish that are caught, obviously by fishermen in tonnes. And then we have the mass in tonnes of the spawning age biomass in the population. So the mass of fish within the population that are old enough to reproduce, basically. So just like with the tables and any graphs you should get, always have a look at seeing the patterns that you can spot to start with. So there's clearly been a dramatic increase in the number of total fish that's being caught every year, which is my black dotted line, massively increasing since the 1950s. And there's clearly a decline, or there's fewer reproductive age fish in the population left. So that's been decreasing, whereas the total number of fish being caught has been increasing. So explain how the graph shows that the population of yellowfin tuna is being fished unsustainably. So remembering what being unsustainable means, it's about not leaving enough for future generations. It's about taking more than can be replaced naturally. So we already have our first and second point, the two descriptions of the patterns that we saw in the graph. And then to get our third mark, we have to then specifically say how those two bits of information demonstrate that it's not sustainable. And it's because obviously not enough fish of reproductive age are being left behind to breed to replace the fish that are being caught. So the number of fish being caught each year is increasing, but the number of spawning age fish is decreasing each year. There are not enough fish being left to breed to replace those that are being caught. You've got some more slightly alternative wording in the mark scheme, but that's the idea of kind of having your two bits of description of the data in the graph, the patterns, and then a link to explain how you understand that that means it's unsustainable. Describe how the mark release recapture method can be used to estimate the number of spawning age fish in the population. So describe how it just wants steps in the process. So steps in the process, how would you actually carry out the mark release recapture method? So we hopefully know this. We've got to talk about sampling, so capturing a sample of the fish, marking some individuals, in this case specifically the ones that were spawning age or young enough to breed, release them, and then resample again to calculate the proportion of marked to unmarked fish in the population that in the sample that you catch, and then that can be estimated up to the population as a whole. We have to remember to describe the fact that we're going to mark them in a way that doesn't harm the fish or doesn't increase their chance of being predated upon. So we're going to collect a sample of fish. We're going to identify the young or the spawning age fish and mark them in a way that doesn't harm them or increase their risk of predation. Release the fish and resample after a long enough time for them to reintegrate. So you don't just like put them back in the ocean and then a second later or half an hour later take them back out. You have to let them reintegrate, and then when you do get that resample, you would count the number of marked and unmarked fish, or count the number of marked fish as a proportion of the sample. In 2004, there was a large earthquake and a tsunami in the Indian Ocean, where the yellowfin tuna were caught. Using the graph and your own knowledge suggests how climate change could have an impact on the future food security of fish stocks. So from the graph, because it says using the graph, we can see there's a massive decrease in the population after 2004. And even up until 2013, that population had not yet recovered properly yet. So once we've taken that information as a result of obviously kind of a large earthquake and a tsunami, but how can we now suggest, using our own ideas, what climate change could have an impact? Could it have a similar impact? And therefore, how could that affect the future of security of fish stocks? And realistically, when we're talking about the future security of food, it's will there be enough to feed a growing population in future generations? So what could cause a similar thing to happen that is similar to an earthquake or a tsunami that could be linked to climate change? So we could think about something like extreme weather, like tornadoes or cyclones. So more extreme weather happening because of that, change in pH of the ocean, change in temperatures of the ocean, potentially leading to migration, so yellowfin tuna migrating or moving elsewhere out of the Indian Ocean, or them going extinct. Anything like that. So we need, for 3 marks, we need a point about what happened in the graph, a point about what might happen, similar might happen as a result of climate change linked and explained, and then how that might affect food security for the future, because there wouldn't be enough to feed a growing population if too many migrate or die. So the graph shows a large decrease in fish population after 2004, which did not return to previous levels. Climate change could cause similar decreases due to extreme weather events. Now we need to make sure we've not just said climate change could cause the same thing, because climate change is not linked to earthquakes and tsunamis, that's geological activity, but we need to link it to something that could cause a decrease in population, an abiotic factor like extreme weather, in order to get that 2nd mark. So fewer fish could be available to feed a growing population. So then that's linking the idea of what we understand by fish stocks or food security. In some places, tuna are caught and farmed in captivity inside large pens in the sea. Explain why these fish grow faster. So it would like reasons why being raised in a pen in the sea or being farmed increases growth compared to being in the ocean. So this is a bit referring back to the energy transfers. So this idea that if you control farming in a certain way by preventing the movement of your organisms too much, they're going to need to do less respiration for the energy to move around, or in case of farming, it would be kind of maintain their body temperature or things like that. So because they're going to not move as much because they're in pens, but also they don't need to go and hunt food because they're going to be given lots of food, that food is obviously going to help them grow faster as well, because they're going to be given plenty of it, lots of it, and be high nutrient food. And also that means there's less respiration required there. So therefore more energy or biomass, whichever you're going to go for here, can then be used for growth instead. So instead of wasting that energy on movement to go and hunt food, or because if they didn't catch enough food, etc., in the wild, then they wouldn't grow as quickly. Whereas if we're controlling that environment, then we're making sure that they're not wasting energy, and more of that energy can then go into their growth, their biomass. The fish in pens cannot swim around as much, and they do not need to hunt prey as food is provided. This means less energy from the food is used by the fish, for movement in this case, so more energy can be used for growth. So I've talked about energy here, but you can talk about biomass if you want to. So more biomass from the food is converted to biomass in the fish. That's fine. You've got sort of slightly different wording and marketing that you can use, but you've got this 4 points, this idea of like they're not moving around, they're being given loads of food, they're not wasting energy moving or catching prey or doing something else through respiration, and so therefore more of that energy or more of their food can then be transferred into their biomass. Okay, so we've got our comprehension question then, which is typical for this paper. It wants us to read the following information and use the information above and your biological knowledge to answer the questions. So now these comprehension questions are not really like comprehension that you get in any other exam, because not really going to be many clues in the question to actually help us answer the questions. It's mostly going to come from our own knowledge. So the first one wants us to define genetic diversity. If we look at line 14, it says it's been successfully collected from many aquatic environments and used to analyse the genetic diversity of remote populations of marine mammals. Define genetic diversity though, just because it mentions genetic diversity in the text, it's not giving us any clues. We have to know what genetic diversity is to be able to define it. So it's the number of different alleles in a population. You've got some slight variation on this in the mark scheme if you didn't quite put it exactly the same as me, but we should know what and how to define genetic diversity. The text isn't going to help us much on that one. Give 2 advantages of eDNA sampling over collecting blood samples from animals. Lines 13 to 14. So sometimes it's going to give you lines that are where something is mentioned, but looking elsewhere might also help you with the question. So this is one of those examples. So it's well documented that DNA is shed from all organisms and deposited as environmental or eDNA, and it has been successfully collected from many aquatic environments. If we look a bit further down, it also mentions on land, eDNA has been successfully collected from feces, snow, soil, honey, and the surface of leaves. So this is why it can really help to read through the whole paragraph before we do some questions, and then you might remember something that might help you with a question that isn't necessarily the lines that they've given you and indicated to you in the question. So this is about giving 2 advantages of sampling this way, or getting DNA this way, rather than collecting blood samples. To collect blood from an animal, you would have to catch them, and then obviously it would be potentially stressful and harmful for you to kind of drug them potentially, and also kind of pin them down or trap them in some way in order that you can actually get a blood sample out of them. With what we're saying here, you can see it's being collected from the environment, from leaves, from snow, from honey. It's not being taken from an animal. You don't need to catch them. Okay, so it's not intrusive. You don't need to catch them. And then we have to now think about a little bit outside the box to think about what we could say is kind of like reasons why this is an advantage. So if you have to find and catch animals, they could be hard to find. You're not going to be completely able to catch them or find them. They could be too dangerous for you to find and catch, like bears, wolves, etc. And then obviously we've said about it being stressful or harmful to the organism. So there are 2 main reasons. So you don't need to track and find all of the animals in order to get their DNA, and it's causing less stress or less harm. It's more ethical than it would be to try and catch them and extract blood. Explain why the Rubisco enzyme is used to identify plants. Lines 10 to 11. In plants, it's the gene that codes for the enzyme Rubisco found in chloroplast DNA. So again, the text isn't much a clue. It just tells us and reminds us that the enzyme Rubisco is found in chloroplast DNA. Only plants have chloroplasts. Only plants have chloroplast DNA. Only plants are going to contain Rubisco because it's needed for photosynthesis to fix carbon dioxide. So really, this is just a do you know what Rubisco does? And we need that word only in there to be making sure that we understand that this can only be found in plants, but it's found in all plants. So it's one of those ones that you can easily make sure by screening quickly that if it's got this enzyme Rubisco DNA code, it's going to be a plant, or it's going to at least be able to carry out photosynthesis. And therefore we can say this is a plant. And the reason all plants will have it is because they have chloroplasts and because it's there to fix the carbon dioxide in photosynthesis. So it's not going to be found in anything else, a fungi, an animal cell, and therefore we can help to rule it out and isolate only plant DNA. Describe the methods the scientists would use to amplify the small samples of eDNA they collect before using them. So what technique or method would they use to amplify small samples of DNA? Hopefully we know that the way we do this, and if we've seen the word amplify, that should automatically make us think PCR, or polymerase chain reaction. What method would the scientists use to extract the specific genes they need for comparison from the eDNA they collect? So it says the most used barcode region for animals. Okay, so it's got mitochondrial DNA and again, the found in chloroplast DNA. So we need to extract, say, the mitochondrial DNA that we're going to use and then extract the specific sequence for the cytochrome oxidase enzyme. So how would we isolate that specific gene from the DNA? There's a couple of ways. You could either extract it using restriction enzymes that have restriction sites either side of the gene that you know you want, or we could use specific primers. If we're doing PCR anyway, we could use specific primers that would only then amplify the gene that we want if we used a DNA-based sequence that was specific and complementary to the sequence just before and just after the gene that we wanted. So we have to say they're specific primers, and we have to mention that obviously they're complementary to the start or the end of the gene that we are looking for. Identify the order of bases in the collected DNA. So what method could they use to identify the order of bases? That's just the same as saying the sequence of bases. So how do we find out the sequence of bases in DNA? It's gene sequencing or genome sequencing or just sequencing. DNA sequencing is fine. That's how you would obviously use the machine, send the sample away, use the sequencing machine to then send you back the sequence of bases in your DNA sample. DNA from chloroplasts and mitochondria is similar to prokaryotic DNA. Explain why these types of DNA were used as reference sequences for the enzymes instead of nuclear DNA. Okay, so this points us to lines 9 to 11, which is again exactly the same that we've been looking at a couple of times, where it's talking about the mitochondrial DNA and the chloroplast DNA. And it's asking us, why did we use DNA from these two organelles as opposed to using DNA from the nucleus? So let's think about some things that it says that's similar to prokaryotic DNA. So what do we know about that? It's short, it will be a circular loop, there'll be no proteins associated with it and no introns. Okay, it's the no introns part that's the thing here. There is a part of the specification that you have to show that you understand that sequencing proteins or getting a proteome from the genome is much easier in prokaryotic DNA because there are no introns. So it's easier to be able to know and find the sequence of a protein, such as an enzyme, and transform that. So maybe take the mRNA and go back to the DNA sequence and then be able to find that sequence, or know that that sequence codes for the protein because there's no introns. So there's no non-coding sections within that DNA. And so we can really easily just go, okay, if this is the sequence of amino acids, these must be the anticodons, these must be the codons on the mRNA, this therefore must be the DNA sequence that is matching that. And then we can go look for that sequence within organisms in order to tell whether they have the cytochrome oxidase enzyme or whether they have the Rubisco enzyme. So it's very much easier, and that's what it's asking us to explain here. So the DNA sequences from chloroplasts and mitochondria would contain no introns, so it would be easier to confirm that the sequence coded for the enzymes. And then it's saying we need to sort of link why not nuclear DNA. So nuclear DNA would have contained introns, and translating nuclear genome into the proteome is therefore harder. So we kind of have to say, why did we choose these two? And then why did we not choose the nuclear to get kind of the full explanation of this answer? This is a hard one. It's taking from a very small part of the specification, but it is something about kind of translating the genome into the proteome that we need to know and be able to explain that it's easier in prokaryotes. Describe and explain how collecting sequences of the same gene over time could be used to determine if natural selection was occurring in a population. So it's saying to look at lines 20 to 21. It's possible that DNA metabarcoding could be used long term to monitor changes in biodiversity, changes in genetic diversity, or even evolution in populations. So what evidence could we get from the sequence of the same gene over and over again that evolution was happening? How could you see it? So think Hardy-Weinberg here, or even just think about the process of natural selection. We'd be looking for a change in allele frequency. So if you have the sequence of the gene and you know the differences between the gene sequence that show you the different alleles, okay, because you'd have to sequence the same gene, look for differences. Those differences would indicate the different alleles of that gene. And then you would have to monitor allele sequences for changes. So if there was a mutation that created a new allele or a difference in an allele, and then that new sequence became more frequent in the population, that could suggest to you that evolution by natural selection was happening. So we would need to compare the base sequences of alleles for the same gene over time to look for differences. And then if the frequency of an allele sequence changes in the population, it could indicate natural selection. So you could talk about here as well, you could say sequence the same gene, look for differences to see or to find the allele sequences, then compare them and then look for changes and see if one of them increases or changes in frequency over time. Again, a tricky question, but this is just about, do you understand how we measure or how we would find out that natural selection is happening? We would expect to see a change in allele frequency. Think about Hardy-Weinberg, and obviously they're saying that allele frequencies wouldn't change over generations unless natural selection or something was happening. So we can look for that. We can do that by comparing the base sequences, looking for changes, and then calculating the frequency of alleles of the same gene in the population over time.
Ouch! This is why in some videos I explain scratches.